Welcome to our alternate month NHIP Advanced Therapies Seminar. So for those of you that don't know, NHIP stands for Newcastle Health Innovations Partners, and it's Newcastle's Academic Health Science Centre. And our Academic Health Science Centre has a number of so-called research satellites, which are entities designed to promote cross-disciplinary research across the NHIP partners. And I won't go into the partnership. You can find all of that online. Before we start, I just wanted to thank Anna Irvin, who's one of our project managers, who's helped to organise this excellent seminar series, actually. And I'm really happy today that Cat Hilkens is our speaker. I've worked with Cat for about 20 years, maybe slightly more now, more or less ever since I arrived in Newcastle. And Cat is a professor of immunology in Newcastle. Shobha, are you going to do the introduction to the seminar itself?
Yes, I can.
Over to you right now. So thanks, Cat, anyway, for joining us today.
Thank you, Kath. It's absolute pleasure to invite you for this seminar series. I'm really looking forward to this talk. Not so fair introduction because I think Kath requires like a very long introduction, but I'll keep it short. So Kath is a professor of immunotherapy at Newcastle University. Her PhD is from the University of Amsterdam. Kath won a EMBO long-term fellowship, a very prestigious award, to work at the Cancer Research UK London Research Institute. From there on, Kath joined Newcastle University to start her own research group. And both in her PhD and postdoctoral work, she became very intrigued by the role of dendritic cells and how they orchestrate immune responses. So she started exploring their therapeutic potential here at Newcastle, and we were very lucky to have her. So at Newcastle, Kath has developed a tolerogenic dendritic cell product that is being used in first clinical trials in rheumatoid and inflammatory arthritis patients. Kath's also one of the founders of the European Network Action to Focus and Accelerate Cell-Based Tolerance-Inducing Therapies. She's also a management committee member of the Arthritis Research UK Rheumatoid Arthritis Pathogenesis Center for Excellence. So here she's going to describe her journey in developing the cell product. And also Kath's a brilliant immunologist. So if anybody has any questions at all, I'm sure she will educate us. Really looking forward to the talk, Kath.
Oh, Shobha, thank you so much for a really nice introduction, and also thanks to
I've been told to mute the video.
Also thanks to Anna and Shobha for inviting me. I'm going to share my screen now and get my presentation going. All right. This sometimes takes a few seconds on my computer. Can everybody see the slides? Is it okay? I hope so. Yeah. Okay, I'll start my timer as well.
See it, Kath.
Great, thanks. Thanks. So yeah, thank you again for inviting me to these seminar series. And as Shobha already mentioned, today I'm going to talk about our journey to develop this dendritic cell therapy for rheumatoid arthritis. So I'm going to talk from the initial concept and all the work that we have done to prepare for 2 clinical trials. So in the outline of this talk, I just wanted to spend a few moments on what dendritic cells are, just a very brief background. Then I'll continue the talk with talking about the preclinical work we've done, the developmental work to develop this tolerogenic dendritic cell therapy. A lot of that is published, so I'll just summarize it. I want to mention some of the work we've done for the tolerogenic dendritic cell clinical trial, so the AutoDecra 1, trial 1 and trial 2. So AutoDecra 1, we actually completed almost a decade ago. And we're now in the process of preparing for the second clinical trial, which will start very, very imminently. So what I also wanted to do is talk a bit about the challenges and the work that we had to do to take tolerogenic dendritic cells from the research laboratory into a GMP facility where these cells can be produced for therapeutic application. And then finally, I'll finish off with some of the things that the future may hold. So first of all, dendritic cells, so they were discovered by Ralph Steinman in the early 70s, and he was really the first person to describe the important role of these cells in the induction of immune responses. And for his work, Dr. Steinman received the Nobel Prize, or was awarded the Nobel Prize in 2011. So key features of these cells are, they're very much professional antigen presenting cells. An antigen is the substance to which an immune response is directed. So they initiate primary adaptive immune responses against incoming antigens. They're also sensors, so they can detect danger in our tissues, so alarm signals from tissues that have been injured or from incoming pathogens. They are extremely potent, and actually one of my favorite mouse paper is a paper that shows that it only takes about 80 dendritic cells to reach a draining lymph node, which is sufficient to initiate a primary immune response. So these cells are very potent, and you don't need many of them. They orchestrate the class of the immune response. So they orchestrate whether the T cells that they're inducing will be mediating a cell-mediated immune response or an antibody-mediated immune response. So they do that as well. And then finally, they play a huge role in balancing the induction of protective immune responses, so immunity, but at the same time also keeping ourselves, keeps the immune system tolerant against our own tissues to prevent the induction of autoimmune diseases. So these are really key features of dendritic cells. And a normal lifespan, if you like, of a dendritic cell is that in the peripheral tissues, let me just get a pointer. So in peripheral tissues, dendritic cells are mostly in an immature state, but in response to incoming danger signals from pathogens or from tissue damage and the tissue factors, they will mature into mature dendritic cells. These cells then have to migrate to the draining lymph node where they will interact with naive T cells. And it is here that they really orchestrate the T cell response by providing T cells with three separate signals. So they provide the antigen signal in the form of a peptide presented by MHC molecules. So this will determine to which antigens these effector T cells will react. They provide a bunch of co-stimulatory molecules, which are really there to potentiate the T cell response, to make the cells going into clonal expansion. And then they also provide signal 3, which is a polarization signal, and that will instruct the T cells in terms of the class of the immune response that they should be supporting. So all these three signals are delivered by dendritic cells to naive T cells in the draining lymph node, and this will give rise to effector T cells that provide protective immunity. But as I've already mentioned, this is not the only thing that dendritic cells do. They can also balance this immunity versus tolerance in the immune system. And it's because of this reason that dendritic cells have gained interest as a therapeutic tool for a range of different diseases. And this is basically because their function is highly plastic. And we have learned how we can manipulate dendritic cells so they become either immunogenic dendritic cells that can support the induction of potent effector T cells that can protect us from a range of pathogens or even cancer. But on the other hand, they can also remain or be induced to become tolerogenic, which means that they will induce regulatory T cells that dampen down and that control immune responses. And because of this plasticity, these dendritic cells have been developed as therapeutics, not only for for the fight against cancer, but more recently also as a tool to treat autoimmune disorders or in transplant patients to prevent the rejection of the transplanted organ. Now, the cancer field is way ahead of the autoimmunity field, and trials with dendritic cells for cancer has been ongoing since 1995, and with some success actually, especially for the treatment of melanoma. Now, the last 10 years, the tolerogenic dendritic cells have been developed for a range of autoimmune diseases, and trials have been actually conducted in the last 10 years, initially in type 1 diabetes, and then in rheumatoid arthritis and Crohn's disease, and more recently also in multiple sclerosis. So work in this domain is very much ongoing. Now, we started 20 years ago, that's quite right, John, that we started 20 years ago with the idea to develop a tolerogenic dendritic cell therapy for rheumatoid arthritis. And as you will probably know, this disease is a very chronic systemic autoimmune disease. It's characterized by joint inflammation and destruction. And the underlying cause of this disease is really a breach in immune tolerance. And this results into unwanted and aggressive T-cell reactivity to autoantigens. Now, there's a range of current treatments available that are actually quite successful in managing the symptoms of this disease to a certain extent. But all these treatments lead to general immunosuppression. There's adverse effects. Some patients become unresponsive. There might be loss of efficacy over time. And the bottom line is that none of these treatments at this point provide a cure. So what we would really like to achieve is to have a new therapeutic that can really induce antigen-specific tolerance, so restore the underlying defect of the disease. And by being highly specific, so highly specifically targeting the antigen-specific immune response, autoimmune response that has gone wrong, this therapy would then have very low toxicity, no adverse effects, and not affecting protective immunity. So that's what we would like. And the question we had was, can tolerogenic dendritic cells offer this solution? Now, when you start to think about developing an autologous dendritic cell therapy as a drug, is that these cells are living. It's a living drug. And so the way that it basically works is that precursor cells are taken from the patient to be treated. In terms of dendritic cell therapeutics, they can be different precursor cells. They can be monocytes, which can be differentiated into dendritic cells. They can be naturally occurring blood DC subsets already present in the peripheral blood, or even CD34 stem cells can be used to drive dendritic cell differentiation. These cells are then taken into the laboratory, GMP facility, and then differentiated into fully functional DC of choice before injecting back into the patient. Now, there's a lot of key considerations when you start to design this kind of therapy, and some of these include, how do you make them tolerogenic? What tolerogenic modification protocol are you using? And how do you assess that these cells actually have tolerogenic function? This is an antigen-specific cell therapy, so you need to load your dendritic cells with the relevant antigen, so there's key considerations there. And then obviously, where should these cells act? Where should they go to? Should they go to the draining lymph node? How do you get them there? Can they still migrate when you inject them? So these are all key considerations that will come up throughout the rest of my talk. And another thing I should mention is that there's different precursor cells that one can use to differentiate therapeutic dendritic cells. Now, historically, we have used monocyte-derived dendritic cells because this was the cell precursor that was historically used for dendritic cell-based therapies. But more recently, there are developments on the way to do similar things with blood-derived dendritic cell subsets or even CD34 positive stem cells. And some of these products have reached clinical trials, especially in the cancer fields. So watch this space. I think this may well be the future. But for the time being, we are focusing on monocyte-derived dendritic cells. And the advantage of using these cells is that we know really well how to work with these cells and how to manipulate them so that we can get the desired dendritic cell phenotype. The other advantages is that it's fairly straightforward to isolate very high purity and large quantity of these cells from peripheral blood products, freshly drawn peripheral blood or apheresis products. Now, we use interleukin-4 and GM-CSF to drive these cells into dendritic cells, and these dendritic cells are immature. They still need to be mature to reach the desired monocyte-derived dendritic cells of choice. Their maturation can be driven by TLR ligands, so toll-like receptor ligands, and pro-inflammatory cytokines and prostaglandin E2. But we can also adapt this maturation cocktail so that we either produce dendritic cells with activating properties, and these would be used for cancer therapeutics. But in our case, we want to have dendritic cells with tolerogenic properties, and there's a range of reagents available, immunosuppressive cytokines or inhibitors of the transcription factors NF-κB. These can be combined with TLR ligands to get dendritic cells with tolerogenic properties. And this is the route we have chosen for the production of our therapeutic TOL DCs. And I should add that because we mature these dendritic cells, they remain very stable. And these dendritic cells are not able to become mature dendritic cells with immune activating properties. So it's a really good protocol to get dendritic cells with stable properties. Now, we have chosen to make our tolerogenic dendritic cells with immunosuppressive drugs, dexamethasone and vitamin D3, that we add at different time points during the dendritic cell differentiation process. The reason for choosing these drugs is because they're effective. They're cheap and they're already being used in the clinic. So making the translation to a GMP compatible product easier. Now, for our TLR4 ligand, we could not use endotoxin or LPS because that would create problems when translating this to the clinic. But luckily, there is a product available that's being used in vaccines, and it's actually the lipid A tail of endotoxin, which doesn't come with the toxicity of endotoxin, but still can activate and mature dendritic cells via TLR4. So we've been using that in our production protocol. Now, like I said, this therapy is an antigen-specific cell therapy, so you need to load the TOL DC with your antigen of interest, and I'll come back to that in terms of what we have been using for our studies. Now, in terms of phenotype, our tolerogenic dendritic cells still have the phenotype of antigen-presenting cells, so they present high levels of MHC class II. They're still able to pick up and process and present antigen on MHC class II, but very similar to mature DCs. But they have a different cytokine profile, and this is probably a very main reason why these cells have tolerogenic function. So they produce very high levels of anti-inflammatory cytokines, whereas their ability to produce pro-inflammatory cytokines is strongly reduced. They also express on their surface other regulatory tolerogenic markers, and I'll come back to that. The mechanism of action of these cells is that they're not inducing the naturally occurring FOXP3 positive Tregs, but they induce another type of T regulatory T cells that are characterized by high production of anti-inflammatory IL-10. And the mechanism of action is partly through TGF-beta. That plays an important role in their mode of action. We've done also work in a mouse model with equivalent mouse bone marrow-derived tolerogenic dendritic cells. And what we could show is that tolerogenic DC loaded with the antigen of interest could significantly reduce the disease burden of collagen-induced arthritis in terms of the clinical scoring of the redness and the swelling of the paws in these mice. And as you can see, when tol DC were not loaded with the antigen of interest, which in this case was type 2 collagen, they weren't effective. So again, this shows that this therapy should be really antigen specific, and you should load your tol DC with the antigen of interest, so the antigen to which you want to induce tolerance. We've also done extensive in vitro studies to understand what these tol DCs would do to human antigen-specific T cells. And in this in vitro study, we sorted T cells that had reacted to a recall antigen, I think it was PPD, in the absence or presence of immature MO-DC or tol DCs. And we then performed a nanostring gene array experiment on these antigen-reactive T cells. And the PCA plots nicely show that the gene expression profiles of T cells that were activated in the presence of tol DC, immature DC, or where no MO-DC were present are very distinct, as you would expect. And when we looked at the gene expression profile of these antigen-specific T cells that had been modulated by tolerogenic DCs, we uncovered very much a regulatory gene expression profile. And all these genes are associated with regulatory T cells type 1, the TR1 cells that were described in 2014. So it shows that, that tol DCs can really push antigen-specific T cell responses towards a regulatory phenotype. We also followed this up with flow cytometry studies, showing that some of these regulatory markers like LAG3, TIM3, and PD1 are upregulated on T cells that are co-cultured in the presence of tolerogenic dendritic cells. And similarly, we also showed that these T cells that are cultured in the presence of tol DCs produce higher levels of anti-inflammatory IL-10 and lower levels of pro-inflammatory cytokines like interferon gamma and GM-CSF. So we were really able to push these antigen-specific T cells towards a regulatory phenotype by our tolerogenic dendritic cells. So all this work then led to a first clinical trial, a safety trial over a decade ago, which is the autologous tolerogenic dendritic cells for rheumatoid arthritis trial, or AutoDecra. I call it here AutoDecra 1 because we're currently working on AutoDecra 2. And what we basically did in this trial is recruit patients with a knee effusion, and we took an apheresis product from these patients and monocytes, and this, of course, this product was taken to the GMP facility, where monocytes were isolated from the apheresis product. We then produced our tolerogenic dendritic cells with all these goodies. And as the source of antigen, we used autologous synovial fluid of these patients, because at that time, we didn't have clear antigen targets that we knew of, but we knew that autologous synovial fluid of patients would include the antigens of interest. We just didn't know which ones they were. This is a photo of real therapeutic tol DCs that were produced. And then after a 7-day production process, this product was then injected back into the patient after in the knee joint, after the knee joint was washed out with saline. And after this, the tol DC product was injected into the knee. And that was the first trial. It was an unblinded, placebo-controlled and dose escalation trial. So placebo-controlled meant that we had patients that received the joint washout but did not receive the tol DC product. It was dose escalation, and like I said, the autoantigen was autologous synovial fluid. We chose the intra-articular route of administration. I can remember that we had long discussions with John about this, because it was the first time that anybody used an intra-articular route of administration. Up to that point, most DC trials would use subcutaneous or intradermal injection. But we thought that intra-articular route of administration would be of interest because it's where the disease is. If it would be safe to use at the disease site, it would give us a very robust safety signal. But also, if something would go wrong and if it would induce a flare in these patients, it would be fairly easy to suppress that flare with an intra-articular steroid injection. Now, although we targeted the diseased tissue itself, we really were hoping that the TOL-DC would migrate to the regional lymph nodes, where they would then instruct naive T cells to become regulatory. But that was a question that we did not know the answer to at that time. The objectives were very much, it's phase 1 trial, so it was very much safety, but also tolerability in terms of the treatment process for the patient, and feasibility in terms of whether we could produce GMP TOL-DC products on a routine basis. Of course, we had a look at efficacy and biomarkers, but this was very much exploratory. The main conclusions of the trial were that intra-articular injection of these TOL-DCs was safe. There were no protocol-defined knee flares in these patients. The protocol was highly feasible. Out of the 10 TOL-DC products that we made, 9 of them passed release criteria. And even the one TOL-DC product that didn't pass the release criteria, we still treated the patient outside of the trial, and all went well. And the protocol was highly acceptable to the patient as well, which was good. So in terms of the effects that we observed was that in the patients that received the highest TOL-DC dose, so 10 million TOL-DCs, we saw that knee symptoms stabilized in 2 out of 3 patients for the duration of the study, which was 3 months. So there seemed to be perhaps some local effects of this therapy in these patients. But when we looked systemically, and we looked at a range of things, we looked at T cell cytokine production, the percentage of regulatory T cells, we looked at serum cytokines, we could find no effects whatsoever of the TOL-DC therapy on these peripheral blood markers. So there didn't seem to be any systemic effect of the treatment. And this raised the question, do these TOL-DCs actually migrate out of the joint? Do they make it to the draining lymph node, which is something that we had hoped for? Now, first of all, I should mention that the in vitro migratory ability of TOL-DCs is not great. So this is an assay where you can measure in vitro the ability of tolerogenic DCs or dendritic cells to migrate in a CCR7 dependent manner. Now, CCR7 is the chemokine receptor that drives dendritic cell migration to the lymph nodes. And as you can see, mature DCs have good ability to migrate in a CCR7 dependent manner. Immature DCs don't have any ability at all. And TOL-DCs are not great. There's some, but it's not as good as mature DCs. But then we also did a real life experiment with one add-on patient to the OTODECRA-1 trial. And we did a study, we did an imaging study with 111 Indian labeled TOL-DCs. And we looked at these, at the signal of these cells at baseline and 20 hours after injection. And basically, there was no evidence from this study that the TOL-DCs managed to get out of the joint and going to the draining lymph node. It seemed that they just got stuck in the knee and didn't go anywhere. So this led to the idea for a second trial, OTODECRA-2. And here we've got a specific hypothesis, which is that for tolerogenic dendritic cells to achieve systemic immunomodulation, they must go to the secondary lymphoid tissue. And so there are some important changes between the 2 trials. in terms of how we set it up and what we're doing, and that's summarized in this table. So because we finished Autodecra 1 over 10 years ago, and when we started to prepare for Autodecra 2, a lot of new information was available on the potential identity of the autoantigens in rheumatoid arthritis. So for Autodecra 1, we used the autologous synovial fluid because we didn't know what antigen to focus on. But now we could make use of highly defined autoantigenic peptides, and these peptides are all citrulinated, and I'll come back to that. So now we had a good target to work with. The dose is different in the second trial because the first safety trial showed that 10 million toll disease was safe, so we decided to go for this dose. But also because in this trial, we're now going to inject directly into the lymph node, we also chose a second dose because we know that we only need 80 dendritic cells to initiate an immune response in a mouse lymph node. I don't know how much we need to induce tolerance, but we felt that by directly going to the lymph node, we should be able to massively reduce the number of dendritic cells needed to initiate the desired response. So here we've got 2 doses we're going to use. The first trial was just intra-articular injection, but now we're going to extend it to intradermal, which is the route of choice for most dendritic cell-based therapies. We still want to compare it to intra-articular injection, and intranodal is actually really a positive control because do we need to get toll DC to the lymph nodes to get the desired immunoregulatory effects? In terms of the samples that we're going to collect, there's a slight change in that in the trial 1, we did perform synovial biopsies, which we're not doing in the second trial, but we're going to get lymph node aspirates. So which will be a really good way to see what's going on in the lymph nodes after injection of toll DCs. Now, the first trial was very much phase 1, so this was safety, tolerability, and feasibility. But in the second trial, which is a phase 2A trial, we really look more at efficacy in terms of correlating the injection route with the immunomodulatory outcomes. So depending on the route of administration, what happens to the antigen-specific, autoantigen-specific T-cell response? That's the question we're addressing in the Autodecra 2 trial. Now, a quick word about why we're using citrulinated peptides as our peptide of choice. It's just because it's a process that's going on in diseases like rheumatoid arthritis. A lot of the autoantigens get citrulinated. So these are autoantigens that contain arginine, which is positively charged. After citrullination, what happens is that this positively charged arginine becomes neutral, and so it almost becomes a neoantigen to which immune responses can be directed. Now, this process of citrullination is catalyzed by enzymes, the PET enzymes. These enzymes are upregulated during inflammation, so this is a process that happens in diseases like rheumatoid arthritis. There's also antibodies that are being raised in these patients against these citrulinated proteins. And actually, these anti-citrulline antibodies, or ACPAs, are now a diagnostic for rheumatoid arthritis and also a marker of more aggressive arthritis. And also, when you look into RA patients compared to control patients, and you look at the number of autoantigen-specific T cells, so T-cell receptors that recognize citrulinated autopeptides. These T cells are enriched in RA patients' peripheral blood and their synovial fluid. And so there's an enrichment of these autoantigen or CIT peptide-specific T cells in these patients. And these are the T cells we believe are pathogenic. And these are the T cells we want to target with our toll DC therapy. So we've got a range of peptides that we're using from a variety of autoantigens. And then a major advantage of this approach is that it also gives us a tool to detect these CIT peptide-specific T cells and to do immunomonitoring on these antigen-specific T cells. So we've got tools in place to see how the functionality of these autoantigen-specific T cells changes after TOL-DC treatment. And we can do that with flow cytometry by using specific MHC peptide tetramers that can detect these CYP peptide-specific T cells. And we can run functional assays like ELISpot assays, where we can look at the production of cytokines in response to our peptide of choice. So it puts us in a good position to do proper immune monitoring in this trial. Briefly, I want to discuss the work that we had to do to get GMP compatible tolerogenic dendritic cells. And this was a very close collaboration with the Newcastle Advanced Therapies unit. And they've been absolutely brilliant because there were a lot of hurdles along the way that we had to overcome. Now, TOL-DCs are, of course, an advanced therapy medicinal product. So there are lots of rules and regulations because the product that we produce needs to be reproducible. We need to be able to define it. It needs to be safe. And of course, it needs to be produced in accredited facilities. So we've got, luckily in Newcastle, a very good GMP cell production facility. And in close collaboration with them, the whole process engineering and validation and quality control of TOL-DC was performed. Now, we had to do a lot of validation and then again validation. And if that wasn't enough validation, there was more validation to do. And this is because the whole TOL-DC production process needs to be validated in the GMP facility. And also you need to set up very good quality control procedures for your cell product. So the TOL-DC product needs to be quality controlled in terms of sterility, viability, purity, contamination, identity. And we also needed a functional assay, a potency assay for the TOL-DC product. All this takes time to set up. And again, this was a very close collaboration with the Newcastle Advanced Therapies unit. To give a few examples, and these are just a few examples that I'm going to give, the actual sourcing and validation of the reagents that we used in the GMP production process caused a few headaches, is fair to say. Some of them could be easily solved. For instance, we decided to change the brand or the manufacturer of our vitamin D3 that we used for the production of our cells. And of course, that was only a small change. It still needed to be validated. And it's time consuming. It can be done, but every time you make a small change, you use a new product, a new manufacturer, it needs to be validated. The MPLA that we use to mature our cells caused a lot of problems. Some of them were fairly easy to solve and others took longer. One of the problems we had, and something I had not realized, being quite naive about what GMP means, is that it was not sterile. And so it meant we had to filter, sterilize it. It then needs to be validated whether it still has the same activity. And then when you use a filter to sterilize a reagent, that filter then needs to go to pharmacy to be tested for its integrity. So you get all these extra steps in the process that you need to incorporate in the final production process. The batch that we had bought from the company precipitated. We never had any problems with this reagent before, but it precipitated, so extra work was done to solve this problem. In the end, sonication of the MPLA was the solution, but again, it needs to be validated whether the activity is the same. And then finally, because we bought our MPLA reagent before the trial started as a big batch, so it would last us throughout the trial, but then we got delayed because of the COVID pandemic and our product expired, which was another headache because the company had decided to close the facilities to make MPLA because they were building a new factory. And so we got slightly stressed. But luckily, in collaboration with the company, they managed to extend the expiry date of our product by doing extra testing. But you can see that all this takes a lot of time to solve. The peptides that we used to load our dendritic cells with our antigens of interest was a very close collaboration with a company in Australia, OSPEP. They needed to test the stability of the peptides, which actually was an issue. One of the peptides oxidized right after production because it contained a cysteine amino acid. That was solved by storing it under argon. We had solubility issues, again, which were solved by producing or changing the formulation of the peptide. So they were made in a different salt form, which was water soluble. To make a long story short, we managed to solve all these hurdles that we came across, but they were just extremely time consuming. And every change that you make, you need to properly validate, and it's time consuming. We also had to do a lot of validation experiments just to demonstrate, for instance, that when you load TOL-DC with your SID peptide cocktail, that it doesn't change their phenotype or their function. So we had to perform those experiments. And also, during the whole process, we decided that we really needed a new quality control marker for our tolerogenic dendritic cells. And this is because of one of the main markers we use to define dendritic cells, or tolerogenic dendritic cells, I should say, was CD83, which is almost absent on most TOL-DC products and which is highly expressed by mature DCs. The problem, however, was, as you can see from this graph, that it's not black and white. There were some TOL-DC products that actually express this marker, and there were also a number of mature DC products that didn't express this marker. So the differential between these two DC products was not black and white, and it did concern us because it could mean that we could end up with a QC of our product that was not okay, or that was not convincing, not black and white enough. So we went to look for other potential markers and tested a range of them. And in the end, we chose to include MERTK as a QC marker for our TOL-DC product. And as you can see, it is quite black and white. It is really well expressed by our tolerogenic dendritic cells. And the other advantage is that it's a positive marker. It's not the lack of CD83, it's the expression of a marker by our tolerogenic dendritic cells that we can use as QC. And the other nice thing about using this marker is that it actually has a regulatory role in the immune system. It's of critical importance in immune tolerance and the suppression of inflammation. So I think that the combination of CD83 and MERTK puts us now in a really good position as good quality control markers for our tolerogenic dendritic cells. We also had to put some work into design and validate a functional assay, a potency assay that we kind of jokingly refer to as an impotency assay, because it's the lack of interferon gamma induction by TOL-DCs in allogeneic PBMCs that we now use as a functional readout for the success of a tolerogenic dendritic cell product. So all this work then led to finalizing the work for the AutoDECRA-2 trial. So after all the validation work in terms of the production of the TOL-DC and the quality control of the TOL-DC was completed, the GMP facility then ran the final 3 successful validation runs. All this work was included in the documents that we needed to submit to the MHRA for regulatory approval, which was granted a few months ago, and patient recruitment is starting really soon. I wanted to tell you another little story, but I've run out of time, so I'll leave that for another time. And I'm going to finish up. What does the future hold? I've already alluded to it. I think exploring other dendritic cell subsets will be of real interest for the future, because I think the naturally occurring dendritic cells have much better migratory ability than monocyte DCs. So watch this space. We could maybe improve the homing of tolerogenic dendritic cells, but I think with the monocyte-derived tolerogenic dendritic cells, you would have to look at gene editing to achieve that. There's closed system technologies under development, which could reduce the cost of producing these kind of therapies. And also, I think maybe combination therapies with toll disease may be advantageous for the treatment of autoimmune diseases. For instance, debulking pathogenic cells and reducing inflammation before trying to induce tolerance. Finally, lots of acknowledgements. This is a huge project, and I probably haven't even listed all the people that have been involved in this project over the last 20 years. There's a huge number of research staff that have been involved over the years with the development of this product. Clinical trials unit have been involved. The Newcastle Advanced Therapies unit have been fantastic, and like I said, it has been a very, very close collaboration with them to get this product to clinical trial. Flow cytometry facility and also our collaborators at the Karolinska have been instrumental. And finally, I want to thank my partner in crime, so to speak, John Isaacs, because without John, toll disease would still be in the laboratory and would not have made it into patients. So I'll end my talk here and ready to and happy to ask any questions you may have.
Yeah, that's fantastic. Thank you so much. So either people can put up their hands or just enter your questions in the chat for Kath. And while we're waiting for that, Kath, can I ask you a question? If that's okay.
Am I still sharing? I stopped sharing.
Yeah, you're still sharing.
Am I still sharing? Let me just stop. There it is. Stop sharing. There you are.
And I've got the chat here. This is really interesting. So, gosh, lots of questions. But the first thing I was thinking is, you know, the toll DC, the DC part 1, do you think the reason your dendritic cells were not migrating is because you kind of already enriched them for antigen specificity? So you grew them with the synovial fluid. And so the chemokine receptor landscape, these,
Yeah, I think you're spot on. I mean, the environment in which we inject these cells is an inflammatory environment, and there will be lots of chemokines that will keep them there. That is definitely an issue. But I think the other issue is that all DCs, monocyte-derived DCs in general, have very poor migratory ability. It's not that none of them make it to the draining lymph node. In the cancer field, they have reported that up to 4% of what you inject can reach the draining lymph node. And that may be sufficient. That may be sufficient. But I think in our case, because we didn't see any systemic effects of the therapy, I think we're quite convinced that they all remained in the joint, and what they did in the joint. We have discussed, and I think we're going to do that in the next trial, is not to inject into an inflamed joint, but actually injecting it in a normal joint, maybe a better route.
Yeah. Yeah, no, absolutely, because, you know, that way you're getting a balance of the chemokine receptors in a normal joint versus, and they can, like, you know, reach the inflamed area as well. So Mark's put a comment in that says, not a question, but a big thank you, Kath, for really giving us a flavor of the end-to-end process. And it really has been a collaborative effort. Looking forward to the trial coming. John's said, it's worth stressing how much the regulatory landscape changed between our two trials. For example, the need for potency markers, it partly explains the time scale. May I ask, what was the reason for this change in landscape?
Well, to be honest, I think already for our first trial, there was already, when we were preparing for that, there was a sudden change in the landscape because of that terrible trial with the anti-CD28 monoclonal that went horribly wrong. Because we thought it would be a walk in the park, and it wasn't. So the first trial was already a shock to the system, and getting that going. But then between the first and the second trial, more and more started to happen. because we had to include new QC markers and assays for the second trial. It was all a bit more professional, if I may say so, for the second trial. Very strict regulatory criteria for the second product. I don't know, maybe people from the GMP facility will be better to discuss that than me. But everything needs to be defined, and you need to prove that it works. And your peptides, the stability needs to be proved. And if it degrades too much, you can't use it anymore. And you're still happy to use it in the research laboratory, but you can't use it in a clinical trial.
Yeah. We've got a question.
Sorry, it was Mark. I was just going to come in there. There are regulatory changes, but there are also trivial things that have an impact. So, even the MHRA becoming short-staffed affects the time frame around their decisions. We've had COVID in the middle where focuses were changed in a particular direction. And as Kat says, sometimes there's another study where a particular regulatory element comes into focus, and then suddenly you're used under greater levels of scrutiny. So it really is an iterative process around developing this and getting that approval. So we can't underestimate just what a positive step it was getting the approval from the MHRA, because I think this was a significant bit of work to validate a bench top process into GMP. So yeah, all good, just about.
We've got a question from NJB306.
Hi, yeah, it's Josh Bennett here. Sorry, it's come up as just my PC name. Thanks for a wonderful talk, Kat. My question was about whether you have a hunch as to how long the TOL DCs will survive in vivo after injection. Just thinking, hopefully, if AutoDecra-2 does show that you're able to induce tolerance, perhaps could we think about how long that would last for, and even whether you'd be looking at repeated injections?
It's a really good question, Josh. So unfortunately, dendritic cells don't live very long. We've done studies in mouse. You can still find them after 5 days, but they're really on their way out. They don't proliferate, so they will just slowly die within maybe a week or something like that. Is that enough to induce tolerance? I don't think so. I think, and we also know from mouse studies already that multiple injections with a product like this would be needed to induce a more sustained tolerogenic response. So I think in this trial, we're not necessarily looking at long-term tolerance induction. Our first question is, which route should we inject the TOL DCs into the patient, and to prove that that can lead to immunomodulation of antigen-specific, autoantigen-specific T cells. Because if we know that, then we've got biomarkers in place that we can then use in future trials where we're going to then probably do multiple injection rounds with these tolerogenic dendritic cells to really kind of look for a protocol that would give us the most sustained tolerogenic modification of the autoantigen response. And it may well be that even then there may be a need for treating these patients at certain intervals to keep that tolerance, to maintain that level of tolerance. We'll see. I also think that the earlier we can treat patients with a product like this, probably the better the outcome will be. And hopefully one day we will be able to go to a scenario where we can prevent the disease from happening at the early stages.
Thank you.
Yeah. And John and Nikki have left a comment, which I will read out. So basically, John says that the landscape's changing because cells were becoming therapeutics in cancer, and the regulators were much more aware. Yeah. Nikki Moyes has added, in addition to the higher QC requirements, the standard of materials, reagents used for therapeutic use, expectations from the regulators have increased, hence changing vitamin D3 to calcitrol, MPLA infiltration, etc. Yeah. So a lot more scrutiny with regards to quality, and therefore work to source and approve reagents.
I mean, in the first trial, I can't remember that we filtered the MPLA. We never did. But yeah, there you go. It was for the second trial, we thought we've been through this already. This is going to be fantastic. We'll do it really quickly. The validation will be easy because we knew what we were dealing with in terms of our product. And then we got, the goalpost had changed. And as already commented on, I think COVID really played a very big role in delaying our study. It has been very, very tough. But we got there. And like John said, getting the approval was very easy. I think the only comment we got is that we needed to identify the person who was labeling the tubes or something like that. I thought they would ask difficult questions about intranodal injections, but no, it was all fine.
That's it. John's got a question for us. John, you're muted.
As usual. More of a comment, really. I mean, first of all, I reiterate Kat's gratitude to the GMP. It's been a journey. It's been a journey for Mark and Nikki and the team, but a really good journey. But I mean, it was just an observation that if you remember, Kat, when we went to the MHRA with Autodecra 1, they asked us what they should be asking us. If you remember, that they asked for a meeting to ask what the question should be because they were so unfamiliar with cell therapies. They had no experience. And I think the difference now is just that there's CAR T cells, there's all sorts of cell therapies out there. And so they've had to develop a much more stringent landscape. So it's...
I mean, the fact that there are more cell therapies is a benefit as well, because during Autodecra 1, we were very scared that they would ask us for safety studies in animals. Because that was the gold standard then, you've got a new product, show your safety in a toxicity model. What do you do when they ask you that question? And we managed to provide the arguments that would not make sense because it's a human cell product. You can't test the toxicity in an animal model. But now that is, it's just not a requirement for these kind of therapies. So in a way, it makes it easier.
Yeah.
But yeah, I think another lesson, what we should be thinking about is, it's very scary to be dependent on one source of material. Like for the MPLA, there's only one company in the world that can provide GMP grade MPLA. And when they stop the production, there's no alternative. And I wish there was a bit more flexibility in the system to then source other reagents and incorporate it quite quickly. But it's the name of the game, isn't it? That's the way it is.
Incidentally, it's worth emphasizing if I wouldn't rule out intranodal as a route of delivery. I mean, if intranodal toll DCs worked, and particularly if it only took a small number of cells, you could imagine developing toll DCs from a blood bag, without all the leukapheresis and that side of things. It could be a very potent product.
Plus then the opportunity to do, to have a treatment scheme where we inject multiple times will be a lot easier if we don't need the 10 million toll DCs. I really think intranodal, you need a lot less than those 10 million. But
Yeah, makes sense actually. Yeah. Okay. We don't have any more questions. Kat, thank you. Can I just thank you so much for this wonderful talk? And if anyone has any questions, I'm sure you can reach out to Kat offline as well. And so thank you so much. Thanks, thanks Anna. Thanks, John.
Thanks, Shobha. And thanks, Kat.
Thank you.
Thanks again for the invitation.
Thanks, Kat. Cheers.
Bye.
Bye.